This video is about the essentials of adaptive immunity, and that is not something that's spelled out as such in these slides. So I'm going to walk you through exactly what I want you to focus on, and then as you go through the slides you will find all of this. But there are parts I want you to focus on now in particular. So part of this is going to be reminding you of things, and part of this is going to be giving you new things. So I want to remind you the innate versus adaptive immune systems are very different, and when we learn the essentials of adaptive immunity, we're looking a lot at how responses are slow, and we're looking at a little bit about how it learns from infection. So those are the themes we're going to touch on here. We're going to see specific immune cells. We will look at B cells, plasma cells, T cells, a little bit of that. And we will use this essential terminology. I will say receptor. I will say display, epitope, and antigen. Probably not display, but epitope and antigen I will definitely say. And it makes sense to jump back to this earlier slide where these things get defined. An antigen causes an immune response, and it can be a protein, but an epitope is what the immune system actually detects, and it's a small fragment of a protein. So your antibody response to the SARS-CoV-2 viral spike protein, so if you get COVID and you have antibodies against that virus particle, it's actually recognizing a tiny fragment of the spike protein, not even the whole spike protein, definitely not the whole virus particle. So that's kind of wild for a lot of people. And I will kind of interchange antigen and epitope because it's more common to see the word antigen, but when people say that, often they mean epitope. So let's get back to it. So this is where we were. One of the things I want to start with is thinking about what is a lymphocyte? A B cell, a T cell. That's what lymphocytes are. So let's see if I can successfully draw that a little bit. What is a lymphocyte? Well, a lymphocyte is a B cell or T cell. That's the general term for two very different types of cells. They look the same under the microscope. All lymphocytes look the same under the microscope, and you can't tell the difference between them, even though there are literally endless varieties of them. So it's a B cell or T cell, and what I want you to think about is what's unique about them. What is so weird about a lymphocyte? Is weird about them. And what's weird about them is their receptors. The B cell receptors or the T cell receptors. Those are the receptors that make B cells and T cells what they are, and it gives them their everything. B cell receptor, T cell receptor, that is what makes a lymphocyte what it is. It's how it sees the world. That one receptor is everything. So let's get into that. So each lymphocyte is covered with 100,000 identical copies of one of those receptors. So if it's a T cell, it's one unique T cell receptor, 100,000 copies of it. Or if it's a B cell, one unique B cell receptor, 100,000 copies of it. So that itself is crazy. They just have one. That means every cell can only see one thing. Each receptor detects one epitope, so every B cell and T cell really specializes in just identifying one specific fragment of one protein. So there's something special I want to show you on the next slide before we finish all this. This here, about how many different receptors are possible, it makes more sense when you look at the next slide, because lymphocytes generate their receptors using a special random DNA recombination event. So early in their development, they take the DNA that encodes their receptors, and in a very specific way, they randomize it. So they do that. They have little mini genes, and they select from those mini genes, and they put them together in new ways to make their receptor. And the possibilities here, when a brand new lymphocyte makes its receptor, there are billions or even trillions of possibilities. And that's where this this number 10 to the 14th comes from. I found this in an immunology book. That's an estimate, but 10 to the 14th power is more than I think that's 100 trillion possible different receptors that a lymphocyte could have. And they get them randomly. So they randomly scramble their DNA to get that receptor. And then it's in their DNA. So they keep making it based on that DNA. They keep making the same receptor, and they can pass it down to their daughter cells. So even though it was randomly generated once, it is more or less fixed from that point on. And so that lymphocyte that has that DNA and all of its daughters will make the same lymphocyte detectors. So if it's a B cell receptors. If it's a T cell receptors. And so again, this means a B cell or T cell is specialized to see one epitope. So that's one fraction of an antigen, just one thing its receptors can see for its entire life. And that is bonkers. So again, well, this brings me to a totally different idea, but it's like the life of a lymphocyte. Well, what is the life of a lymphocyte like? A lymphocyte only has one sense that matters. It's like it has a sense of smell. It has an olfactory receptor, just one, but it has 100,000 copies of it. And it doesn't have any way of knowing what its receptor will detect. And we have no way of knowing what its receptor will detect because its receptors are randomly generated. And they're random. We don't know what they detect. And so lymphocytes just sit there with other lymphocytes in your lymphoid tissue, in your lymph nodes, and in your other lymphoid tissue. They just sit there waiting to see if they're ever going to smell the thing that they're going to smell. And by smell, I mean if their receptor will actually detect the thing it can detect. And they don't know if they will. Most lymphocytes never detect the thing that their receptors can detect. So this is fundamentally what makes it different from the innate immune system. Innate receptors are coded into our DNA at birth, and we all have the same pattern recognition receptors. We all have the same toll-like receptors. And we know exactly what they detect. But you cannot say that about a B cell receptor or a T cell receptor because they are randomly generated. And so every lymphocyte is unique. And that gives us this drawing here. This drawing is of 3 B cells, and I can tell that because from the shape of the receptor. And this is a great thing to draw when you study adaptive immunity, is force yourself to draw 3 lymphocytes and draw their receptors. And what I want you to notice is that this receptor is a Y-shaped protein with a binding site, with 2 binding sites, basically. And this is the same receptor, this is the same receptor. And the binding site here is shown as being rectangular. So the idea is it would detect some rectangular molecule. Whereas this one has 3 copies of a different B cell receptor that would detect some like curved epitope. And this one has 3 copies of a different receptor that would detect some very pointy epitope. So practice drawing that. And those are B cells. T cells, if I draw a T cell, it's going to look like this. It's a circle, and the T cell receptor is going to look like a rectangle with a bite taken out of it. It's very different from a B cell receptor. And this is literally what you see in immunology textbooks. They have these goofy drawings with these rectangles with bites taken out of them, or a B cell, again, which is just a boring circle. Under the microscope, they are totally boring. There's nothing to see. But it's going to have these Y-shaped proteins with binding sites on the end. And the way I'm drawing it here this B cell and this T cell recognize the same thing. They both see this curved semicircle. And so the epitope they're looking for, I'll try to draw the epitope they're looking for in beautiful galaxy, because that is what I am now. They're looking for a circle. Whereas the ones in the slides might have been looking for a little triangle or a little square. And those shapes are just what we use to remind ourselves that a receptor detects a molecule because their shapes match, like a key going into a lock. They have to have a shape that matches. And so the receptor is the lock, the ligand or epitope is the key. So that's what is a lymphocyte. And that is something you need to remember. Memorize this slide. Understand what's in this slide. I cannot overstate how important that is. So, okay, that's what is a lymphocyte. The second thing I want to talk about is lymphocyte development. That's what I want to talk about next. And basically, I'm going to give you a list of steps they're going to go through. And I want you to copy this down and memorize this backwards and forwards. Because lymphocytes go through this developmental process, and you need to know it before we can understand immune responses. So let's give ourselves some room here. So they start as, oh cool, galaxy, immature lymphocytes. Whether they are B cells or T cells, they start as immature lymphocytes. And they go through a process of education. If they survive that process, they become mature but naive lymphocytes. This drawing pad is not what my other one is. It's a bummer. This education we'll talk about in a lot more detail. It is a selection process, and 95% of immature lymphocytes die. So the 5% that survive are mature but naive lymphocytes. They wait. They hang out and wait. Mature but naive lymphocytes wait to see if they will go through activation. And if they go through activation, which is rare and only happens during an immune response, then they become active or activated lymphocytes, or we could just call them B cells, T cells, something like that. There's no clear terminology that I know of for them in this state once they have been activated. But there is terminology for what they do in the next thing they do, and that is clonal expansion. And that is where they divide as fast as they possibly can. They make as many daughter cells as they possibly can in a short period of time. And each daughter cell is a clone. They're making perfect copies. This is asexual reproduction. They are perfectly copying themselves. So they're making clones. So they're expanding their population by making clones. So clonal expansion. And then their daughter cells have two possibilities. They can become effector cells, or they can become memory cells. So effector cells are the ones that are going to do the immune response. And I'll tell you next what the effector cells are and what they do. But the memory cells are the ones that wait for the next exposure to this pathogen. So they are what gives you your immune memory. Your memory cells are activated cells. They're the daughters of activated lymphocytes. But instead of going off and fighting and doing an immune response, they wait for the next time. So what a vaccine is supposed to do is activate some of your mature but naive lymphocytes, and they'll go through clonal expansion. You'll get effector cells, but you'll get these memory cells that will wait. So if you've had a vaccine, the idea is you'll get memory cells, and then when you actually get the disease later, the memory cells kind of start over and go through an activation process again, and they give you huge numbers of effector cells, basically. So that's lymphocyte development. Next, we'll look at effector cells. What are the effector cells? There are 4 different effector cells that we're going to learn. So let's get into that. I jumped ahead to this slide because it has everything sort of written out. The effector cells are here on the left. On the right, we have different things. We have mature naive cells, activated cells, dendritic cells, which we talked about in innate immunity, and then memory cells. So those are that. These on the left are effector cells. So what you need to do here is pause this video and write down all 4 of these and practice, learn, memorize the heck out of these 4. You need to recite this in your sleep, every part of it. So CD8 positive cytotoxic T cell, what's its function? It kills abnormal host cells. CD4 positive helper T cell type 1, what is its function? It tells macrophages to focus on intracellular pathogens, or generally to be more aggressive. So in immunology books go back and forth about what exactly signals they're getting, and in some cases, the macrophages just get more aggressive, it sounds like, and in others, they focus on intracellular pathogens. So bacteria they ate through phagocytosis that they haven't killed for some reason, they get a signal to kill those. The helper cell type 2, CD4 positive helper T cell type 2, these are the ones that give activation signals to B cells. And then plasma cells are the ones that make antibodies. So those are the 4. So let me clarify these 2. Let me just write these in a less formal way. So I'm going to give you the helper TH1 function. And you can go back and see what a TH1 is. But a TH1 function gives be aggressive signal to macrophages. That's the informal way I'd write it on the board if I were doing this lecture face to face. So that's what helper T cell type 1, CD4 positive helper T cell type 1, that's what it does. A CD4 positive helper T cell type 2, or a TH2, gives activation signals to B cells. And what you'll see is that leads to B cells making effector cells, because that's what activation of a lymphocyte is. It leads to effector cells and memory cells. And specifically, the effector cells for B cells are plasma cells, and they make antibodies. That's their job. And so the next and final part of this long video will be, what are antibodies and what are their functions? You know what? Never mind. This video has gotten way too long, so I'm going to instead make a separate video just about antibodies. I think that will make more sense. Yeah, okay.